Welcome to this episode of Eureka, and we are going to have a very interesting and informative conversation with Dr. Anil Suri, Director of National Institute of Immunology, one of the autonomous organization under the Department of Biotechnology. By the by, about 7.5 lakh people die every year out of cancer in India, and almost 1 lakh die out of cervical cancer. Whereas about 86% of cancer patients get themselves screened early enough in Western countries, it's just about 12.5% in India, which makes cancer a dreaded disease in our country. We are going to discuss quite a lot about cancer, how to prevent it, what they are doing, what's in offing. But before we start our conversation, let's take a quick look at his brief profile. Keep watching Eureka.
Cancer is the leading cause of death worldwide. Dr. Anil Suri is among India's outstanding translational cancer researchers who are promoting our healthcare system by taking his discovery from benchside to bedside to treat cancer better. Dr. Anil Suri is currently the Director of National Institute of Immunology at New Delhi. Dr. Suri graduated from Hansraj College, Delhi University. He did his post-graduation and doctorate from NDRI Karnal. Subsequently, he completed his postdoctorate from LNCR, France, before returning to India in 1985 and joining NII as a scientist. Dr. Suri's thrust area in cancer research is mainly on early detection and diagnosis of the disease. He has developed targeted approaches for therapeutic interventions for cancer medicine. Dr. Suri discovered the novel cancer antigen called SPAG9 that is crucial for the earlier detection and diagnosis of reproductive tract and breast cancer. His breakthrough in cancer immunotherapy provides precision and personalized treatment for cancer patients. Dr. Suri's discoveries led to clinical trials on the first ever cancer vaccine developed in India for cervical cancer patients. The phase 2 trials are underway at the Cancer Institute, WIA Adyar in Chennai. They are being conducted by Dr. T. Rajkumar, MD Oncology, on 54 cervical cancer patients. Dr. Suri authored 63 high-impact works in reputed international journal along with 15 patents on novel immunotherapeutic method for cancer treatment and diagnosis in India and abroad. He has been honoured with the prestigious S.S. Guraya Memorial Award, Dr. T.C. Anil Kumar Memorial Oration Award, Rabindranath Chakravarti Citation Award, besides many others.
So thank you for being with us. It's a wonderful opportunity. It's a very important and I suppose one of the major health issues in the emerging India. I think infectious diseases are taking backtrack because of our public health intervention. So cancer is becoming one of the major cause of death in India. We need to look at it very seriously. Let me start with one question which is there in our mind. You know, when we were, let's say, school kids or college kids, we used to hear about cancer only in film. If in the plot of a story, you want to kill one of the heroine, so that the hero can marry the other heroine, you give this cancer to that second heroine and you kill and then create a melodrama. But today, most of us know at least one person in our close circle who has died out of cancer. Why this change?
You rightly pointed out. Basically, what's happening is because of our variety of reasons are responsible for what we are seeing in today's scenario. And one of it is the lifestyle, and another is our aging, which is actually also contributing. As you know, by 2020, we'll have an average age of Indian population will be 29. And because of that aging process and lifestyle, which includes environmental factor, your living styles, your eating habits, your smoking, drinking, everything counts, which can contribute to the onset of oncogenesis. But having said that, it is also important that we must address and bring in that we have now more of medical infrastructure facilities which can detect cancer and then treat the cancers. But unfortunately, as you rightly said in the beginning, our patients walk in into our clinics at very late stages where the treatment modalities options are very limited.
Yeah, that's a thing. Let me come to another issue. Your major work has been in cervical cancer, particularly finding a vaccine for cervical cancer. I want to explore this story. But before that, I want to understand what the cervical cancer is. How does it happen? I mean, one can understand that how, let's say, for example, a oral cancer can occur because of substance abuse. Maybe you are smoking, maybe you are using gutka pan, which is one of the major cause for cancer burden in India. Yeah, but cervical cancer, I mean, it's just our part of our natural body structure. How does it occur?
Well, if you consider in low and middle income countries, this is a major problem, and for that reason, for our country, it is one of the major problem. As you said very rightly in the beginning, that over 1 lakh patients die because of cervical cancer, and it's because of our most of the percentage of population, around 70% population, lives in the rural areas. And this is cervical cancer onset is because of an infection of human papilloma virus, and 95% plus patients or women get transmission of this human papilloma virus because of unhygienic conditions, because of malnutrition, because of diet, and because of their poor health. They get infected when they are reproductive, active, and this infection onsets. And basically, HPV virus, what it does is it gets integrated with the genome of cells in the cervical cancer.
Oh,
cervical
so it means like the virus itself enters into our cell.
Right.
And masquerades as our normal gene.
Absolutely.
Okay.
So there are checkpoints for cellular proliferation, which is actually set right from the beginning of our conception when the baby was formed, from an zygote to the embryo and to the fetus and to the adult body. So we have check balances where we have tumor suppressor genes which make tumor suppressor proteins. So what these human papilloma virus does is it doesn't allow the tumor suppressor protein, which is one of the protein which is called P53 protein, and doesn't allow that protein to in fact work properly so that the cells can not turn into a de-differentiated mode.
Okay, let me try to understand this. What you are saying is that the tumor is nothing but when the cell multiplies uncontrollably in an abnormal way. But usually we have some kind of a gene which actually says that no, you are doing too much, stop. But this particular virus is able to even stop that monitor. Is that so? I mean, it's like police himself is becoming a thief.
Exactly. So what's happening here is the checkpoint balances within the cell cycle of a cell division. It interferes into the cell division, and then the cellular proliferation begins. And then that's how, when the cellular proliferation begins, initially the body actually try to mount its immune response. The cells will try to overcome if there is any abnormality that they see. If you understand human body, every day around 50 to 70 billion cells will die. I mean, guess where do they go? And how do they die? These are all cells which are programmed cell already. So they have to die. They perform.
It's a very natural stuff.
That's a very natural functioning of the cells. But guess what? If the cells are not allowed to die, then there is going to be a kind of the formation of the cells which will evade, which hijack your nutritions, they hijack their blood supplies, and they make their own environment to survive, and they evade immune system, and that is how it starts growing. But initial stages, early stages, if one can detect, 90+% patients can be cured 100%.
Very interesting. And you said a very interesting point. I mean, we think that suppose if we can live forever, I think that will be the bliss. But then if a cell decides to live forever, that's actually causing the cancer, right? That's a very interesting.
Yeah. So it's saying something like it will never die.
Yeah.
It is immortal.
Immortal.
Yes.
So immortality is not necessarily good at cellular level.
Absolutely.
Very interesting. We'll take a very short break. We'll continue. I mean, we are going to have a very interesting exploration into the world of cancer, what we can do, what their institute is doing, and what he has done. After the break, keep watching Eureka. Welcome back to Eureka, and we are having a very interesting and very engaging conversation with Dr. Anil Suri, Director of National Institute of Immunology, an autonomous organization under Department of Biotechnology. Before the break, we were discussing about cervical cancer, which is a very huge burden in India, particularly to women. And your work has been on that, and we also talked about how cervical cancer is due to an infection by a virus. So, what's your exact work? I mean, how did you get interested in this whole area, first of all?
I mean, to begin with, ever since I started my career, I was very much fascinated by this particular disease, and I was very inquisitive about how these cells can actually, they are the most corrupt cells in the body, as we discussed earlier, and they can modulate, evade immune system. So I was very inquisitive about how these cells can, do all the changes within living in a normal body, but be abnormal for the body and cause problem eventually and cause the death. So the discovery, the research ongoing program was that I initiated my exploration, and I knew that it cannot be done in the institute, so I have to have an outreach program. So that's how my journey started, to reach out to the hospital. I call this as a reverse translation. So I wanted to go into the clinic. What is the problem that they have? What is exactly going on in the clinics? And how the patients are walking in, and what stages of the cancer they are walking in. And I found out that the stages that they were walking in were not treatable at all, even with the chemotherapy available at that time. And I always say this, wherever I have, and I loudly say that in my discovery of this whole journey of this molecule, the key partners were patients. So the patients actually gave the tissues. So what initially we did was, what I did was, I wanted to see if we can find some signature for these cells. Signature means something specific for these cells that are expressed in the cancer cell but not in the normal cells. Way back 30 years ago, we didn't have a state-of-art facilities which is available right now, like differential gene expression, micro DNA, microarray chips, where we can do the subtraction. But at that time, we had to do everything manually. So I did a subtractive hybridization library. I made it. What I mean to say here is that I collected all the messages that are transcribed in the cancer cells, and I collected all the messages which are transcribed in the normal cells, and then I minused it. So I picked up those messages which are exclusively expressed in the cancer cells.
You use the tissues collected from patients and then normal cells, and then you found the difference. Then what happened?
Okay, so then we started, when we found this different signature, then we pick up those messages, we cloned them, and then we try to express the protein. Now, it is always that when you look at the transcription, it is not necessary that it would be translated protein as well. Now, we wanted to make sure that it gets translated. For that, we again had to go back to the cancer tissues to look into the expression, whether it is expressed or not. So we generated antibodies against these antigens in the animals and used it, and that is what the whole journey began. That these patients were expressing whopping, there was a huge expression within the cells of these molecules, and whereas in the normal cells, we didn't find any expression at a protein level. So the validation, your validation starts now, then if you see a protein in the cell, you should understand that these proteins will be performing a function for a proliferation.
Good. We'll take a very short break. We'll continue this story. It's a very interesting story where at the end of the day, they have come up with a vaccine which perhaps is going to save lives of millions. We'll take a very short break. We'll continue this conversation. Keep watching Eureka. Welcome back to Eureka, and we are having a very wonderful and a very engaging conversation with Dr. Anil Suri, Director of National Institute of Immunology, an autonomous organization under Department of Biotechnology, and he is involved in discovering a vaccine for cervical cancer. There is a mythology in South India particularly where Kartikeya, the son of Shiva, is supposed to have been the teacher of Shiva. I am told there is also a very interesting story like that in your life. We want to hear it.
Okay, so what happened was, this is a story of a child going to a school. She was, I think, 17 years back. She was in 3rd or 4th class, and her class teacher actually asked the question, please raise your hands whose parents are engaged as researcher or they are scientists. So none of the kid raised their hand except the child. And then she said, okay, stand up and tell what are your parents doing? So she says, my father actually is engaged in doing cancer research. And so teacher asked what exactly he's doing. She said he's trying to find some molecules for cancer patients for their cure. And then teacher replied, what if he fails and he doesn't found? Then his whole life will be wasted. And the child didn't understand, and she said, and the child happens to be my daughter. And now she's pursuing a medicine. She's in America, in a hospital. She came back home and she was like 3 days, not bubbly as she used to be. And I was like kind of curious. I just asked her, what's going on? And everything fine, your school is going well. She said, no. I said, do you want to talk to me something? I said, yeah, I want to talk to you something. I want to ask you something. I said, yeah, ask. She says, guess if what you're doing, if it doesn't work, your whole life will be wasted.
Yeah, you searched for something all your life and then you didn't find anything at the end of the day. Perhaps your life is wasted. Is it so?
So, no. So I immediately smiled and I put my hand on her head and I said, no. I said, we learn from our failures. Your good judgment will always come from your failures. We'll have 9 failures and then 10th will be success. Same thing happens in the research. We can't leave it. So if there will be 10 people working on this cure, for a cure for a cancer, 9 will fail, 1 will succeed. So we always learn from our failure. Our failures contribute to our success. So go back to your school and tell your teacher that no, even if he fails, there will be others who will be working on a similar lines, and definitely we'll have a cure. And here we are today, that discussing about how the discovery came through. So this is
interesting stuff. I mean, you are one of the 10, not the 9, at least perhaps the remaining. So what is your discovery? What is this vaccine? What does it do?
Actually, this cancer vaccine that we are having right now is basically a dendritic cell based vaccine. And I have a collaborator, it's a medical oncologist team at Cancer Institute Adyar who are doing these clinical trials in collaboration with us. And this particular molecule that we have is basically is called SPAG9, which is used to prime the dendritic cells and educate the dendritic cells and then inject back into the patient. So basically it's a personalized intervention for a patient itself. So they have a very detailed protocol inclusion criteria. If the patient is expressing the SPAG9 protein, then she is included in the clinical trial. and then her peripheral mononucleoside cells are harvested, and they are ex vivo grown in the culture, and they are primed with the SPAG9 early premature dendritic cells, and then after 10 days 3 to 4 days growth, they make these doses from 1 million, and they inject back to the patient
the same cells.
The same cells.
Cells taken patient, from you.
From the patient, taken from the patient. So 0, 14, 20, 1, and then month, and then monthly. So almost 13 patients they have done in their clinical trials. So basically, this is Dr. T. Rajkumar who is undergoing these phase 2 clinical trials in
So what is this state in it? I mean, I'm told that there are different phases in which all these things have to be tested. So where is this right now?
Yeah, so basically for drugs that you use for a normal human bodies, the phases of clinical trials can be defined differently. But since we are talking about the cancer, in cancer we have the patient who are clinically terminally ill patients, and in this particular case we are into phase 2 clinical trials. That means they have the stage 3 B cervical cancer patient, where it has actually cannot be resected, or it can be only treated with chemotherapy or radiotherapy. And phase 1 clinical trials were also conducted way back in 2006 in Cancer Institute, Adia, Chennai. That is, they had used the tumor lysate. So basically, you can either use the tumor lysate as well of a patient, because it's expressing the same protein. So in that trial, they had used the tumor lysate, but your phase 1 clinical trials involved tumor lysate to see whether it is having safety or not, right? And phase 2 clinical trials, they have now comparing tumor lysates of the patient, of the same patient, one arm, and the second arm is with the SPAG9, which is a purified recombinant protein, and treating the patient, other patients as well. So they are comparing tumor lysate versus SPAG9 expressed protein. So phase 2 clinical trials, number of patients increase, and it is safety and efficacy. The phase 1 clinical trial is basically a safe.
So can we say that phase 2 clinical trial you have come up with colors?
Sorry?
Or is the phase 2 clinical trial is still going on?
It's still going on. It's all going on.
So what will be the next step?
So then we will have to go into phase 3 clinical trials. Basically, you have to have expand the number of patients from 100 to 3,000, and you have multi-institutional hospitals involved, multi-centric involved. And then phase 4 is the post-marketing that you compare with.
So which means that you are at kind of, let's say, a big breakthrough. Suppose if this phase 2 comes up with flying colors, which means that we can really have a vaccine which can cure millions of patients. Is that right?
Yes, this is what I must give credit to my collaborator, Dr. T. Rajkumar, who is actually a medical oncologist, and these trials are basically conducted by the clinical researchers, and his team, oncology team, pathology team, they are all doing very hard work, employing the molecule that we have discovered here, and it is basically a dendritic cell based vaccine therapy. Definitely this will go in a larger volume.
Though we don't have much time, I want to ask this question because it's very, very important. I mean, if you look at the statistics from Europe or US, for every 100 cancer patient, 60 to 65 survive for 5 years, but whereas that figure for India is just 20. So what do we need to do? Where do we lack?
I think to begin with, we must have the awareness programs. And awareness, I mean that we must educate right from the schooling to undergraduates to the postgraduates about how to go about this cancer preventive oncology. And second is that we do, as I mentioned in the beginning of the program, that around 70% of our population lives in the rural areas. So there also we need to have primary health care centers, where initially we should have the pap smear at least done to prevent this one of the most deadliest cancer which is actually taking toll in India, which is cervical cancer, which can prevent more than 95% of our population of women who is dying because of cancer. And another thing is that there are other institutions also involved, where within our own institute also, where we are trying to find out genomic signature sequences for predispositions of the cancer, and genomic landscape of the whole cancers, a variety of cancers. And certainly we need to upscale our medical infrastructure and facilities and have the outreach program in place. That is very important.
Thank you. Thank you. It's been a very wonderful and very engaging, very informative conversation. Keep watching Eureka. We'll come back with another conversation next week. Keep watching Eureka.